Have you ever wondered how your cuts heal or how babies are made? This is our presentation to answer your questions. Presented to you by Nate Antonik, Ryan Decker, and Jared Spurlin. But what happens if these processes take a turn for the worse? Dun dun dun! Let's start with mitosis. First comes synthesis with the replication of DNA. The nucleus dissipates. Then the chromosomes line up on the metaphase plate. Next, the spindle fibers pull the chromatids to polar sides of the premature cells. The cleavage furrow starts to appear, and the two nuclei are created. Finally, the cells split, creating an identical daughter cell. Thus, your wound is magically healed. Sex cells go through a different process called meiosis. Meiosis goes through 2 cell divisions rather than 1, resulting in 4 haploid daughter cells. In prophase 1, the chromosomes begin to condense. Crossing over occurs in this phase. The purple and red non-sister chromatids exchange DNA segments in our model. Metaphase 1 brings the tetrads to line up at the middle of the metaphase plate. In anaphase 1, pairs of homologous chromosomes separate, one chromosome moving toward each pole. In telophase 1, each half of the cell has a haploid or single N set of chromosomes. Simultaneously, cytokinesis is when the cell splits into 2 daughter cells. Animal cells form a cleavage furrow, and plant cells form a cell plate. Meiosis II is very similar to the process of mitosis, except that in meiosis II, the DNA does not replicate beforehand. After the process of meiosis, 4 haploid daughter cells remain. They can either be sperm cells in males or ovum cells in females. However, the processes don't always go as planned. A mutation is a change in the genetic material of a cell or virus. Mutagens are physical or chemical agents that can cause mutations. Point mutations are chemical changes in just one base pair of a gene. A single nucleotide in a DNA template can lead to an abnormal protein. There are 3 types of point mutations. Base pair substitution replaces a nucleotide and its partner with another pair of nucleotides. Insertion, which is the addition of a nucleotide pair. Likewise, a deletion is the loss of a nucleotide pair. Frameshift mutations are caused by insertion or deletions. They code for different unplanned RNA and proteins. Nonsense mutations change an amino acid codon into a stop codon. This leads to non-functional proteins. Missense mutations code for an amino acid, but not necessarily the right amino acid. Polyploidy is a condition resulting from meiosis in which an organism has more than 2 complete sets of chromosomes. This includes triploidy when there are 3 sets of chromosomes, and tetraploidy when there are 4 sets of chromosomes. Monosomic and trisomic zygotes also result from meiosis. A monosomic zygote has only 1 copy of a particular chromosome, while a trisomic zygote has 3 copies of a particular chromosome. In non-disjunction, the pairs of homologous chromosomes do not separate correctly during meiosis. As a result, one gamete receives 2 of the same type of chromosome, while another doesn't receive a copy. Aneuploidy is when an offspring has an abnormal number of a particular chromosome. Aneuploidy results from the fertilization of gametes in which non-disjunction occurred. Mutations found in mitosis differ greatly from those caused in meiosis. Proto-oncogenes are the normal cellular genes that are responsible for normal cell growth and division. Mutations to proto-oncogenes cause oncogenes. Oncogenes are cancer-causing genes. Dun dun dun! Oncogenes code for more cyclin proteins. Cyclin proteins bind with cyclin-dependent kinases, or CDKs, creating MPFs, or maturation-promoting factors. These MPFs allow the cell to pass the checkpoints in the cell cycle. When you have too many cyclin proteins created by the oncogenes, the number of MPFs rise, allowing the cell to repeatedly pass the checkpoints, thus sending the cell cycle out of control. The out-of-control cell keeps multiplying. This leads to excess cell clumps called tumors. Another name for these tumors is cancer. This biology documentary has been brought to you by Ryan Decker, Nate Antonik, and our narrator, Jared Spurlin. Thanks for watching.